Hello, grade 12 students. In this video, we will cover the genetics exercises on your book. So you have to open your book and you have to follow us during this video. First, let's start with probing the documents on page 69 with the first question. Here we have the main types of point mutations and their consequences. We have the non-transcribed DNA strand, which is non-transcribed, is called the non-template, it's the coding, and gives a quick and direct reading of the messenger RNA, mRNA, by just replacing thymine T with the uracil U. So it's very easy. You have only to replace the T with U. You replace thymine with uracil so that you can get the mRNA. So the first type of mutation we have substitution, we have deletion, and we have insertion. Deletion and insertion, they cause something is called frame shift mutation. Okay, whereas substitution is of three types, either missense or silent or nonsense. But the meaning of missense meaning that it codes for another amino acid. Whereas silent mutation, there is no change in the sequence. While nonsense, it would lead to the stop codon. So let's start step by step in the first one. So in the first one, we have the sequence CCA, CAG, and ACT. If I want to find out the transcript, this is the non-transcript strand of the normal and their corresponding amino acids. So if I want to find the sequence of the transcript, which is called the template, and it's the non-coding, so I have to make the complementary. The complementary of C, it's G, and the complementary of C, it's G, the complementary of A, it's T. And here also the complementary of G, it's C, the complementary of A, it's T, and the complementary of G, it's C. And also here the complementary of A, it's T, C, it's G, and T, it's A. This, if I want out to find the complementary strand of the non-transcribed, here it's called the transcribed. Also, if I want to find out the mRNA, as they said, we can find out the sequence of the mRNA by only substituting thymine with uracil. So the sequence of the mRNA will be as follows. It will be CCA, here GAG will stay GAG, and ACT will be ACU. This is the mRNA. These will give us the protein Pro, GLU, and THR. In the mutated one, in the mutated sequence, we have CCA, and here we have GTG. So here, the second nucleotide of the second codon. So this is the first codon, this is the second codon, and this is the third. So this is the second nucleotide of the second codon was substituted. So we have here the same amino acid Pro. Here we have GLU, here we have VAL, valine. So we have different in the amino acids. So change in the second nucleotide, in the second codon, where A is substituted by T. This leads to change in the amino acids from GLU to VAL. So this is called a missense mutation. While in the second substitution that causes silent, substitution in the third nucleotide in the second codon, G was substituted by A. So that give the same amino acid sequence. So there is no detectable change in the sequence of the amino acid. So the formation of protein won't be altered. So this will lead to something called silent mutation. Whereas the third type of mutation is called the nonsense. It goes to the appearance of the stop codon. While deletion, here deletion, we deleted one of the nucleotide. So deletion of one of the nucleotide leads to something called frame shift. So here we have TAC, ACC, ACG. TAC, it remains the same. We deleted the A, so it becomes CCA, here CCA. And then we have CGA, CGA. So we deleted one of the nucleotide. This will lead to something called frame shift point mutation by deletion or either by insertion. So TAC, TAC. Here we insert a nucleotide, here we inserted the G. So it becomes GAC, GAC. Then we have CAC, CAC, and finally we have G and A, G and A. So this is called frame shift. So here the first sequence where we have CCA, GAG, ACT. This is the non-transcribed, the non-template. It's the coding. While this is the complementary, which called the transcribed, the template, the non-coding. CCA, GAG, ACU, mRNA. We can get it into two ways. Either it's the same as the non-transcribed, where T is replaced by U, or we can make it complementary for the transcribed, and the T is replaced by U, where G the complementary is C, G is the complementary is C, T is the complementary is A, G, C is the complementary is G, T the complementary is A, C the complementary is G, T the complementary is A, G is the complementary is C, and A the complementary is T. And mRNA we don't have T, we have U. So this is the amino acid sequence. And in the mutated we have a difference in one of the amino acid, so this is called a missense. So a change in the second nucleotide of the second codon, where A is substituted for T. This coded the change in the amino acid sequence where GLU is substituted for VAL, for valine. Silent mutation, as we said, silent mutation, it leads to the same amino acid sequence where third nucleotide of the second codon was substituted for A. So this leads to no change in the sequence of the amino acid. So this is called a silent mutation. So formation of protein won't be altered. While for the deletion, we deleted one of the nucleotides where A is going to be deleted, and then we have the sequence, same amino acid before, and then they change here. So this is called frame shift mutation. By what? By deletion. So moving to programming the documents on page 66, let's talk about the first part. We have DNA molecule. This DNA molecule is cleaved by two different restriction enzymes. First one is called EcoR1 and the second one is called NOT1. Before we start, we have to write the formula. You have to know that the number of fragments, it's equal to the number of restriction sites. It's equal to the number of restriction sites plus 1. This is the initial formula and this is what we have to work on. So number of fragments is equal to the number of restriction sites plus 1. Here they want to find out the number of restriction sites. First of all, let's talk with note 1. Note 1, how many fragments do we have? We have 1, 2, 3, and 4. So we have 4 fragments. While in the EcoR1, how many fragments do we have? We have 1, we have 2, we have 3, 4, 5, 6, and we have 7 fragments. So here we have 7 fragments. So we have the number of fragments. This one is known. So we want to find out the number of restriction sites. So number of restriction sites, number of restriction sites is going to be number of fragments minus 1. Since plus 1 comes here. So number of fragments minus 1. This is the answer here. So let's start. Note 1, how many number of restriction sites? Number of fragment 4, so it's 4 minus 1, so 3 restriction sites. While in the EcoR1, how many fragments do we have? 7. So number of restriction sites 7, number of fragments minus 1, so it will be 6 restriction sites. So let's talk about this. So recognition site of the echo R1 is repeated six times. Since echo R1 cuts the DNA into 7 fragments, whereas recognition site of NOT1 is repeated 3 times. Why? Because NOT1 cuts the DNA into 4 fragments. You have to use this formula. Number of restriction sites is equal to the number of fragments minus 1. Now let's continue with parts 2a and b. This electrophoresis of 2 different alleles, they are cleaved by the same restriction enzyme. It's called BAMH1. They said explain the results obtained in document C. What causes this difference? So here you have the restriction site for the BAMH1 is GGATCC. It's repeated 1. GGATCC is repeated the second time. GGATCC is repeated 3 times. So here we have cleavage. So here we have the work of the allele 1. It shows 2 band after being cut with the BAMH1, since the recognition site is repeated 3 times. As you know, the number of fragments, it is number of restriction site plus 1. So here it's repeated how many times? 3 times. And then we have allele 2. Allele 2 results from a mutation. Why? Here we have GGA-TCC and here we have GGA-TCC. Right, but here G is replaced with A, so we have here a mutation. So allele 2 shows only 1 band. So this mutation is caused this appearance of the one of the recognition sites here. So the allele is not cut from inside. So 1 band is observed. Here the one is called the C. It's near to the negative pole. So near to the negative pole, meaning that it's the heaviest one. And we will talk about this in the coming slides. Here we have A, B, and C. So band A, this is called a band. From now on, we call it a band in the gel electrophoresis or in the zemogram. This is called a band. Moving now to the answer. So allele 1, it shows 2 bands after being cut with the restriction enzyme BAMH1, since the recognition site is GGA-TCC. Is repeated how many times? 3 times. Allele 2 results from mutation to allele 1, and this mutation causes appearance of 1 of these restriction sites. So the allele is not cut from the inside. So 1 band is observed, indicating that we have only 1 fragment. Referring to document D, classify. This is document D showing the gel electrophoresis. Classify the fragments of the DNA of document C according to their decreasing length order. Justify your answer. As you see here, band C is closer to the negative pole than band A, than band B. So here you have to know that DNA represented by band C migrates less than one represented by the band A, and less than that represented by band B. Okay, so from here you have to know that the DNA of the band C is much larger than the DNA of band A, and also is larger than that of band B. So note that the thick band, it represents 2 alleles. So you have to know from now on, thick band, it represents 2 alleles, and these alleles said to be the homozygous individual. So band C is closer to the negative pole than band A, than band B. So DNA represented by this band C migrates less than that represented by band A, less than that represented by band B. So DNA of band C is larger than A and larger than B. This note is very important, that the thick band, it represents 2 alleles and the person is said to be homozygous. Now let's move to the exercises on page 72. So pay attention here to these exercises. Open your book on page 72 and follow me. Exercise 2. They use the genetic code table to answer the following. this genetic code table, you have to use it in order to answer the following. What is the amino acid sequence of the peptide translated from the following DNA strand? Part B, how would the deletion of a nucleotide at position number 5, this is position number 5, affect the sequence of this polypeptide? Justify your answer. So here we have the sequence TAC, TAA, CGG, then we have ACA, TCA, and ATT. This is the sequence of the DNA, and this is the template strand. Okay, this is the template transcribed, and it's the non-coding. So if I want to find out the sequence of the mRNA, first of all I have to find the complementary. So the complementary of T, it's A. The complementary of A, it's T, but we should not put T in the mRNA, we should put U. So let's start with the solution. Also, the sequence of the mRNA will codes for the sequence of the amino acid. So amino acid, I don't want to study them for sure, so you have to find them out using this genetic code table. So here we have TAC, TAA, CGG, ACA, TCA, ATT. This is the DNA, the template, the transcribed, the non-coding. If I want to find the mRNA, I have to make it complementary. So the complementary of T, it's A. The complementary of A, it's T, but you have to put U. C, it's a G. T, it's A. A, it's U. A, it's U. C, it's G. G, it's C. G, it's C. A, it's U. C, it's G. A, it's U. And T, it's A. C, it's G. And A, it's U. And also A, it's U. T, it's A. And T, it's A. So we have the sequence of the mRNA. Sequence of the amino acid, MAT, isoleucine, alanine, CYS, serine, and stop. New DNA sequence, we have a mutation. What is this mutation? We deleted the 5th nucleotide. So we deleted the 5th nucleotide. So the sequence will be as follows. TAC, TA, the 5th nucleotide is deleted. So we have frame shift. So we have TAC, GGA, CAT, CAA, and then we have TT. So we have the sequence of the amino acid MAT, PRO, VAL. So the mutation causes the disappearance of the stop codon. So the stop codon disappeared here. So this causes a change in the amino acid sequence, which becomes larger. So there is no stop codon, and it will continue its way. So this will lead to the increase in the size of the protein. Now let's move to exercise 5. A mother thinks that her child was exchanged for another at birth in hospital. This occurs a lot, by the way. Document below represent a DNA fingerprint performed to find out the truth. Here's the father, and here's the mother one. Here you have the child. Has her child been really exchanged for another? Justify your answer. This is called the restriction map. This restriction map of the father, or let's say with the child and the mother. Mother and the child, we have a common band. This common band here between the mother and the child. So DNA of the child matches the DNA of the mother, and this which should be the biological mother. So the child wasn't exchanged. Also, it shows DNA with the father, a common bands with the father. So the mother chose a common band with them, so she is the biological mother. So restriction map of the mother and the child show a common band. So DNA of the child matches the DNA of the mother, which should be the biological mother. So the child hasn't been exchanged. Moving to exercise 6. Exercise 6, talking about the sequence. The sequence TCT AAA TGG is one of the possible sequence of the DNA coding. They gave you this non-transcribed, non-template, it's the coding. So if you have to find out the sequence of the amino acid is the same, where T is replaced by U. This corresponds to 3 different amino acids. What are the different types of gene mutation? All of you know that we have either deletion or substitution or insertion or addition. Part B, how can you obtain silent mutation in this case? Silent mutation is substituting one of the nucleotide with another but having the same sequence. In this case, we have TCT, AAA, TGG. So here we have silent mutation if it gives the same amino acid sequence. Identify the possible point mutation involving only one base, which lead to the stop codon. If you want out to the stop codon, you have to replace one of the codon leading to the stop codon. So here we have 3 kinds of point mutation, either insertion, deletion, or substitution. This is the non-transcribed, the non-template, it's the coding, TCT, AAA, TGG. If you want to find the transcribed, so complementary. And if I find the amino acid, the mRNA, so TCT, so I make it the same. Instead of T is replaced by U as shown here. And here the T is replaced by U as shown here. So this will cause a silent mutation, whereas the second non-transcribed, the transcribed TCT, AA, TGG, this will lead to the same amino, and this will lead to the stop codon UGG, UGU. Okay, so this will lead to the stop codon. Exercise number 7. Pay attention to this exercise. Exercise number 7, showing a bidimensional chromatography is performed using a mixture of different human amino acids. The chromatogram of figure 1 is thus obtained. The figure 2 chromatogram showed the results of tetrapeptide hydrolysis of an individual X, whereas the figure 3, this is figure 3, it shows the chromatogram showed the results of the hydrolysis of the same tetrapeptide of another individual Y. Pay attention to this because it's a little bit critical. First, what is the molar mass of the tetrapeptide shown in figure 2? As you see here, you have to know that tetrapeptide as shown here. So tetrapeptide, you have to compare it with the control sample. This is the control sample. So the first one is, this one is ASP. We compare them according to their position. And this one is for ALA. This one for LYS. And the last one is for VAL. Okay? So if I want to find out the molar mass, how can I find it? It's found here. So ASP, this is aspartic acid. This is 133. If I want to find, for example, lysine, LYS, this is 146. If I find out alanine, ALA, it's 89. If I find out valine, VAL, it's 117. So here we find out the molar mass of the tetrapeptide. So I have to add 133. First of all, 133, which is for aspartic acid. And you have to add 89. You have to add 117 for valine and 146 for lysine. It will be 485. This is the molar mass, 485. Part B, they said, can one determine the exact sequence of this tetrapeptide? Justify your answer. Exact sequence. Can we find out the exact sequence? Exact sequence? No, we can't find out this exact sequence. We find out that we have these amino acids. We have aspartic acid, we have lysine, we have alanine, we have valine. But I don't have the sequence, how they are arranged. How many types of combinations do we have? We have different combinations. How many? We have 4 different amino acids. So it's 4 factorials. It's 4 times 3 times 2 times 1. This is called 4 factorial. Why I choose 4? because we have 4 different amino acids. 4 factorial, it's 4 times 3 times 2 times 1. 4 times 3, it's 12. Times 2, 24. Times 1, so it's 24 different combinations. Part C, how can you explain the chromatogram of individual Y? The chromatogram of individual Y, it shows 3 bands, while that of X here in figure 2, it shows 4 bands. So here we have 4 bands, while here we have only 3 bands. So one of these is missed. So we have aspartic acid, since it's the same position. We have lysine, LYS, and we have alanine. So the one missed is here is the valine, VAL. This is the one missed. Okay, so this maybe can be explained. It's code for a mutation, and maybe the valine, nucleotide, the nucleotide that code for valine, they are substituted, or we have a mutation in this nucleotide and they code for a stop codon. That's why we don't have valine. Okay, so how can you explain this? We have 3 instead of 4. Maybe the sequence is mutated, and this mutation leads to a stop codon and prevents the formation of the valine amino acid. So the chromatography diagram 2 shows 4 bands corresponding to 4 amino acid: alanine, aspartic acid, valine, and lysine. So the molar mass, we added them, it's become 485. How did I add them? You are given in the question. Part B, they said, can we find out the sequence of this tetrapeptide? Sure not, because we have only the name of these amino acids, so we can't find out the sequence. We have a lot of combinations. How can we find out the combination? It's 4 factorial. So from now on, this is called factorial. Exclamation is called factorial. This is in math. You got it in math, I think. So 4 factorial, it's 4 times 3 times 2 times 1. So it's 24 different combinations. Part C, how can you explain the chromatogram of individual Y? Individual Y, we said it shows 3 bands instead of having 4 bands. So one of these bands is missed, which is valine. So the amino acid valine is missing from the tetrapeptide. We can explain this, that the fact that the last codon in the DNA is code for valine in the normal case. So this codon will become a stop codon and valine will disappear. So now let's move to exercise 8. The next document is obtained by using a synthesis radioactive mRNA and the corresponding DNA strand in the case of certain proteins. After we interpret this document, you have to show how certain gene mutations might have no effect on the phenotypic expressions. This diagram is showing DNA. Some of them, they hybridize, as shown here, they hybridize with the mRNA that's shown in the, let's say, in the yellow or the brown color or the orange color, while others, they don't hybridize. So fragments of DNA, they have no complementary on the mRNA. These, as they mentioned them, they are the non-coding regions. Okay, so DNA or the gene in general contains something called exons and something called introns. Okay, introns and exons. Exons, they bind to the mature mRNA, while the introns, they don't have complementary. So we have to work with the exons. Introns, they are going to be eliminated. So if any mutation happens in the non-coding region, these mutations will not lead to any consequence. So will not lead to any problem in the sequence. So if this mutation occurs in the introns, these ones that are going to be eliminated, so no mutation in the mature mRNA, and these introns, they are going to be eliminated. So if you have mutation here in the introns, these introns will be eliminated, so no effect on the messenger RNA. For that, some mutations, they are referred to as silent mutations. Why? Because they don't have any effect. So any mutation in the intron will code nothing, and this is called silent mutation, or it's called neutral mutation. Why? Because these entries are going to be eliminated. So the diagram shows the parts of the DNA buckles don't hybridize with mRNA. So the fragments of DNA have no complementary on the mRNA. So DNA or the gene contains exons and introns. Both are translated to give immature mRNA that contains exons and introns. What will happen? Before its export to the cytoplasm, mRNA is subjected to processing. Introns, they are cut and eliminated, while the exons, they are joined together to form the mRNA, which is the mature mRNA. Exons of DNA, they bind to this mature mRNA. Buckles, they represent the entrance of the DNA that have no complementary in mature mRNA. So the mutation that takes place in these entrants, it may not lead to any mutation in the mature mRNA, since these entrants will be eliminated. So these mutations, they are referred to as silent or they are coded neutral and they have no effect. That's why we have something called the silent mutation, since it affects the entrance. Okay? Thank you.